All right, let's study your gluteus maximus off. Seriously though, every single time you smile, blink, take a breath, or go lift a heavy weight, there is this incredibly intricate, totally hidden world of cellular machinery firing away inside of you. Today, we're diving right in to demystify exactly how our bodies move.
Welcome to The Explainer, the anatomy and physiology of a muscle flex. Our goal here is simple. We're going to take some notoriously dense molecular biology and translate it into a relatable, highly visual story about human movement, so these complex concepts actually stick with you.
Here's our roadmap for the day. It's a structured zoom in, zoom out journey. We've got 1. The muscle blueprint. 2. Anatomy of a flex. 3. The sliding filament model. 4. A neuromuscular love story. 5. Measuring the magic. And 6. Exercise is medicine.
Let's kick things off with Section 1, the muscle blueprint.
So our bodies actually run on 3 main classes of muscle cells. You've got smooth muscle handling things behind the scenes, like wrapping around your intestines. You've got that beautifully striated cardiac muscle that's completely exclusive to your heart. But our focus today, skeletal muscle. Notice how it's made of these elongated, multinucleated fibers running perfectly parallel. The defining feature here is that it's electrically excitable and completely under our voluntary control. It connects to your bones via tendons to let you actively navigate the world.
Moving right along, let's dig into Section 2, anatomy of a flex.
The spatial breakdown of a muscle is basically a biological masterpiece. Think of it like a giant set of Russian nesting dolls. An entire muscle breaks down into smaller bundles called fascicles. Pop open a fascicle and you find individual muscle fibers. Those are the actual cells. Inside those fibers are tightly packed cylinders called myofibrils. And if you zoom all the way in and pull out a single myofibril, you finally hit the tiny myofilaments. These are the proteins actually doing the heavy lifting. Which brings us to the sarcomere. This is the fundamental repeating microunit of the muscle. This is the VIP room where the real magic happens. It's defined by these boundaries called Z-discs, and it houses our thick filaments, called myosin, and our thin filaments, called actin. The physical shortening of a muscle happens right here, inside this tiny geometric space. All right, pop quiz time. Before we get into exactly how they interact, let's test your focus. Based on the structures we're unpacking, which of these specific proteins combine to create that complex thin filament? Are we talking actin, myosin, troponin, tropomyosin, or maybe a combo? Take a quick second to lock in your guess. And the answer is E. If you guessed it, you nailed it. The thin filament isn't just one single thing. Actin forms the main structural chain. Imagine these really beautiful, braided, bead-like molecules. But it doesn't travel alone. It has bodyguards. Tropomyosin is this string-like protein that physically covers up the active binding sites when your muscle is just chilling at rest. And troponin? That acts as the biological lock. It's a specific site where calcium is eventually going to bind to kick off a contraction. Now let's see these proteins in action with Section 3, the sliding filament model. Watch this dynamic closely. See how those thick, pink myosin filaments reach out, grab the thin blue actin filaments, and just pull? The absolutely wild thing here is that the actin and myosin do not change their own length at all. They literally just slide past one another. That sliding action pulls those outer boundaries closer together, shortening the entire sarcomere. I want to emphasize this because it's absolutely crucial. As the sarcomere shortens and those Z-discs at the edges get pulled inward, the dark A band, which is defined by the full end-to-end length of the thick myosin filament, keeps its exact length. It stays completely constant the whole time. The only thing that changes are the zones of overlap as the thin filaments are dragged toward the center. So, what's the spark that ignites this whole process? That brings us to Section 4. The source material literally calls this a neuromuscular love story. Every movement you make starts with a motor unit. It's this incredibly intimate physiological partnership where a single somatic motor neuron, reaching all the way out from your spinal cord, connects to and directly controls a bunch of individual muscle fibers. A rapid-fire electrical command from your brain blasts down this neuron to wake those fibers up. Let's break down this communication sequentially, because it is a flawless chain reaction. First, an electrical action potential hits the nerve terminal. This forces calcium channels open, which basically tells vesicles to dump a neurotransmitter called acetylcholine, or ACH. The ACH floods across the tiny gap and docks at the motor end plate of the muscle. This binding throws open channels that let sodium just rush right in, depolarizing the membrane and sending an electrical wave surfing all the way across the muscle fiber. Next, that electrical wave dives deep into the muscle tissue, signaling the sarcoplasmic reticulum to release its internal floodgates of calcium. Remember our lock from earlier? This internal calcium binds straight to troponin, which physically yanks that tropomyosin string right off the actin binding sites. The targets are exposed, myosin immediately grabs on, forming a cross bridge. Then, burning a molecule of ATP for energy, the myosin head executes the physical power stroke, pulling the actin and shortening the muscle. Boom, flex achieved. Let's pause for a slightly morbid but honestly fascinating interactive check. Based on this very specific chemical dance we just talked about, what exactly causes the absolute rigid stiffness of rigor mortis when a body stops functioning? Why do the muscles permanently lock up? It's because there is no ATP available. As we just saw, the initial power stroke uses ATP, but here's the critical catch. A brand new, fresh molecule of ATP is physically required to bind to the myosin head just to make it let go of the actin. When you die, your body's ATP factory shuts down. Without it, the cross bridges are permanently locked in a sustained flex, and that's what causes the intense stiffening of rigor mortis. All right, back to the living. Let's talk about Section 5, measuring the magic. Using a tool called electromyography, or EMG, we can slap surface electrodes onto the skin and essentially eavesdrop on these exact myoelectrical signals. By capturing these distinct waveforms, doctors and scientists can measure neuromuscular activation in real time. We are quite literally watching the invisible electricity of your motor units firing. So looking at those electrical waves, how do our bodies actually generate more force when we need to pick up something super heavy? Think about it for a second. Does a faster electrical signal or a fundamentally stronger overall signal change the tension of the muscle versus the recruitment of more fibers? Well, here's the crucial distinction. Firing a single motor unit faster and faster just ramps up the localized tension within that specific unit as the twitches start piling up on each other. However, increasing the overall stimulus strength to the muscle, that calls in backup. It activates entirely new additional motor units, a process we call recruitment. More recruitment equals greater overall active force from the entire muscle working together. Which brings us to the big picture takeaway. Section 6. Exercise is medicine. The source materials sum this up beautifully. Exercise is literal medicine. By routinely waking up and activating these sliding filaments, you aren't just looking good, you're actively improving cardiovascular endurance, regulating your blood glucose, and physically alleviating stress, anxiety, and depression at a chemical level. Knowing that every single flex is this incredibly complex, beautiful cellular symphony that heals and protects your body, what are you going to do today to fire up those filaments?